Hello and welcome to today's session. Our topic for today is microorganisms and human diseases. Microorganisms and human diseases. So far, we have been able to go through the introduction of microbiology, where we talked about the history, the prehistory of microbiology, and then the history of microbiology. Talked about modern microbiology, where the germ theory started and how it got confirmed. And then we have talked about organisms, classification of organisms, talked about the processes that are used in classifying and identifying organisms. Now, today we are going to look at microorganisms and human diseases, the diseases that are as a result of microorganisms. Now, let's look at the introduction of this topic. Robert Koch believed and proved the germ theory of disease. Robert Koch's postulates forms the basis for determining the specific human diseases are caused by specific microorganisms. Now, remember we talked about Louis Pasteur, who started the germ theory, but unfortunately he died without being able to prove the germ theory. But then this man called Robert Koch came up later on, a German scientist, and then he was able to confirm this theory, that's the germ theory, by cultivating or growing an anthrax bacteria, the bacteria that causes anthrax, in mice, and they started to cause the infection in mice. And that was how science was able to confirm that diseases are specifically caused by organisms or microorganisms. The ability of a microorganism to cause disease is known as pathogenicity. Pathogenicity. And then pathogenic microbes have virulence factors. So an organism causing disease, or for the ability of an organism to cause disease, we refer to this process as pathogenicity. So a pathogenic organism is an organism that can cause diseases, whereas non-pathogenic organisms are organisms that do not cause diseases. Okay. And I'm saying that pathogenic microbes, or microbes that cause diseases, have gotten certain things we call virulence factors. These are the factors that enables them to be able to cause diseases. So let's see. Virulence factors are special properties that enhance the ability of pathogenic microbes to cause diseases. So virulence is the degree of pathogenicity. That is how pathogenic an organism is, how disease causation or disease causative the organism is. Example: colonization and ability to grow within host cells, ability to overcome immunity of host, ability to produce toxins. All these factors are virulence factors. If you have an organism that is able to produce toxins, that organism has a virulence factor. For instance, you might kill the organism, but the organism might have already produced toxin. The toxin is going to cause disease for you. So that is a virulence factor. The ability of organism to grow in an environment that is hostile is a virulence factor. Okay. Infectious diseases progress through contamination, colonization, infection, and diseases. So what we're saying is, diseases that are infectious, or that we refer to as infectious, can actually move from one person to another, or infect a person through contamination. So the person will have to first get contaminated with the organism, and then the organism will colonize the person. So let's say the person uses or touches a fomite that has gotten some organisms on it, and the person touches the nose with the hands. Now, the nose are contaminated, so the mucous membrane of the nose will get contaminated with organism. So, the organism now colonizes the mucous membrane of the person. Then, it will lead to infection. The organism will start to multiply, and then they start to cause the infection or the disease. Then, finally, the disease results. Now, let's look at infection and disease. Infection is the growth of microorganisms within the body of the host. So for we to say infection, we mean the organism that causes the disease has actually gotten access into you and it has started growing in you. Disease is the disruption of normal body function. So you've gotten an infection, and then that infection has led to your inability to be able to function well as you used to. For instance, so you've gotten the disease called pneumonia. The infectious agents have gotten into you, they have infected you, and they have led to the disease called pneumonia. So once you've gotten pneumonia, the lungs are no longer able to expand and contract like they used to be in order to exchange oxygen for carbon dioxide. So as a result of this, your respiratory system becomes compromised. So that means you have developed a disease. Half of all human diseases are caused by infections. Let's look at infectious or communicable diseases. Infectious or communicable diseases. These are diseases that can be passed from one organism to the other by way of droplets, fomites, and contact. So for we to say an infection or a disease is communicable, it means that these infections can actually be passed, or the disease can be passed from one person to another, or from one organism to another organism. This could be by direct contact, by droplets, by fomites, etc., etc. Epidemiology of a disease. When you say epidemiology, what are we talking about? Epidemiology is the study of the frequency, the distribution, and the distribution of diseases and the factors which contribute to their spread. So when we say somebody is studying epidemiology, or the course the person is offering is epidemiology, the person is studying diseases. He is looking at the distribution of that disease. How is the disease distributed? What category of people are getting it the more? Which part of the world, or which part of the environment, let me say, which part of the environment or the world is the disease much more common? What category of people, which time of the year is the disease common? So all these factors, if you are studying them, we mean you are studying epidemiology of diseases. When we say epidemic disease, what are we referring to? Diseases that are constantly present in a community. So we have certain diseases that are constantly present in a community. Almost all the time you have people who are suffering from that disease. So if you take a place like our environment, like Ghana, like this, or sub-Saharan Africa, a disease that is almost always constant, like people almost always have it, is a disease like malaria. So malaria is an endemic disease in our part of the world. Then we have epidemic disease. Epidemic. This is occurrence of diseases much more frequent than usual. So when we say an epidemic has occurred, then we mean there is this disease that we know of has been happening, but it has a time came and the disease has occurred much more frequently and abundantly than it used to be. So in that case, we call it an epidemic. So sometimes they have epidemics like meningitis or cholera, which sometimes will come at a period within the year, and a lot of people will die, and then within a period of time it vanishes again. So that is epidemic for you. Then we have pandemic disease. Pandemic. This is a worldwide epidemic of a disease. So when we have an epidemic, a disease occurring in excess of its usual ranges, and this disease has spread to all parts of the world, then we call it a pandemic. An example is the current situation we are facing with the COVID-19, which we call the COVID-19 pandemic. Then we have sporadic diseases. Sporadic diseases. Diseases that occur occasionally. So once a while, you have the disease happening. Once a while, you have the disease happening. That is what we call sporadic diseases. let's look at patterns of disease. Patterns of disease. Specific characteristics that characterize diseases are called signs and symptoms. So when we say the signs and symptoms of the disease, then we are referring to the specific characters that distinguish this particular disease from the other disease. All right. So signs of a disease are observable through examination. So when we say we want to distinguish now between signs and symptoms, signs are the manifestations that you can observe through examination. So like say you touch somebody and the person is warm, that is a sign. You check the person's temperature and the temperature is high, fever, it is a sign. You see somebody vomiting, it is a sign. You examine somebody and you see that the tummy is distended or the person has edema. These are all signs. Now let's look at symptoms. Symptoms can be observed and felt only by the patient. So the symptoms are the ones, the manifestations that it is only the patient can feel them or observe them and tell you. For instance, a client that is nauseous, he feels like vomiting, but he has not vomited. So that feeling to vomit, that is nausea, is a symptom. It is not a sign. And then, like say the person is having abdominal pain, the pain is a symptom. It's not a sign because you can't see it and there is no way you can elicit it. Combination of signs and symptoms is called syndrome. So when we say the clinical syndromes of the disease, then we are referring to the combination of the signs and the symptoms of the disease. The after effect of a disease is known as the sequelae of the disease. So the sequelae is referring to the aftermath of the disease. What happens? The consequences or the final consequences of diseases. Now let's look at stages of infectious diseases. Stages of infectious diseases. We have incubation period. That is the time between the entry of microbes into the body and the onset of symptoms. Organisms adapt and multiply to cause infection during that period of time. So the incubation period talks about the time the organism entered into your body and the time you start to show the signs and symptoms. So there are certain diseases that have incubation period very shortly, like within a few hours, like 12 hours, you start to manifest with the signs and symptoms. Whereas other diseases will take very long time, like weeks, like months, even sometimes years, before you start to show the signs and symptoms. So the period from when the organism entered till the time you start to manifest your signs and symptoms or your clinical syndrome is what we refer to as incubation period. Now let's look at prodromal stage. Prodromal stage. First signs or symptom of disease which are non-specific and occur and are highly communicable. I mean, the disease is highly communicable during that period of time. So what we are trying to say is, when we say prodromal stage, we are talking about after the incubation period, when the person starts to manifest with the signs and symptoms, they may show some signs that may not even be specific to the disease condition. For instance, you are having a condition like cholera, you just got an infection, and then when the incubation period is up and you want to start to manifest the signs and symptoms, you may first show a sign like headache or some mild fever, which may not be specific to the classical signs of cholera that we know to be profuse vomiting and diarrhea. So these early signs that you see, or the first signs that you see, are what we refer to as a prodromal stage. And at this stage, the disease is very, very communicable. Period of illness. Specific symptoms develop peak of signs and symptoms. So what I'm trying to say here is, during the period of illness, the person has started developing specific signs and symptoms of the disease. And that is the peak of the clinical manifestations of the disease. Now, period of decline. That is the convalescent stage where first signs of recovery appear and symptoms abate. So during the convalescent stage or period, we have, or during the period of decline, we have the client gradually recovering from the disease. Full recovery, that is the end of the disease syndrome, and patient returns to full health. All right, so let's look at types of infections. We have primary infection. That is an initial infection in a previously healthy host. So let's say one is free or had never suffered from coronavirus infection, and unfortunately the person had it for the first time. That is all referred to as primary infection. Secondary infection, this occurs due to the depletion of host defenses by the primary infection. So let's say the primary infection was able to break you down, it was able to break down your immunity, and as a result of that, it is either the disease worsened or you have other disease that are related to that particular disease coming on board. We call that secondary infection. We have acute infection. These develop and complete their course rapidly. So acute infection, when we say an infection is acute, it means that the infection develop and complete their full course rapidly. Whereas chronic infection develops slowly, less severe, and they last longer than acute infections. Subacute infection, this is an intermediate condition between acute and chronic infections. So it is not as short, it doesn't happen abruptly and completes its course shortly like acute infection, and does not also last longer as chronic infections. That is what we mean subacute infection. Then we have localized infections. These affect a circumscribed area of the body. So a localized infection will always be localized or be residing on only a part of the body without affecting the general system. Then we have generalized infections. These affect the entire systems of the body. Let's look at what we call chain of infection. Chain of infection. A series of events that happen in order for an infection to occur is what we refer to as a chain of infections. So what are these series of activities or events? For an infection to develop, each link of the chain must be completed. Breaking any link of the chain can stop the transmission of the infections. So when you go forward and then you get to know the specific events, what we are trying to say is, if you are able to take off one of these chains or one of these events, then you stand a chance of stopping the infection from completing its course. So we have the chain of infection here. You can see in the middle top we have the agent, we have the reservoir, then we have the portal of exit, then we have mode of transmission, then we have portal of entry. All right, so we have the agent here, then we have, this is the agent that actually will cause the disease. Then this is the reservoir where the organism is based or the agent is based. Then the reservoir has gotten a portal of exit. Portal of exit is the route through which the agent will exit from the reservoir. Then when the organism is able to exit from the portal of exit, then there should be a mode of transmission by which the organism that has exited from the reservoir can now enter into the susceptible host, and that's what we call the portal of entry. So the organism enters the portal of entry in the susceptible host, and the susceptible host is the person who stands a chance of getting the infection. So let's take for an example, give an example to see if we can illustrate this. Agent should be Vibrio cholerae, the organism that causes cholera, and the reservoir is, let's say, food, some contaminated food, say rice, that was contaminated or is contaminated with the agent, which is Vibrio cholerae. So this is a bowl of rice. Then Vibrio cholerae, which is the agent, is in the bowl of rice. Now, there should be a way that this organism will exit, and that is by, say, the person eating the food. So the person will use a spoon, fetch the food from the reservoir, and then the mode of transmission is through the fecal-oral route. So whatever the organism in the spoon of rice will now have to enter into the person's mouth. That is mode of transmission, fecal-oral route. Then entering into the person's mouth is the portal of entry, and then the person who is eating it is a susceptible host. So this person has finally eaten the food, and then eventually the person develops cholera. All right, so that is it here. The infectious agent, we have the reservoir, portal of exit, we have mode of transmission, we have portal of entry, and then we have the susceptible hosts. Okay, so I have already explained this, but if you take the infectious agent, that is the disease-causing organism is known as the etiologic agent or the infectious agent. Example of these are bacteria, viruses, fungi, protozoa, chlamydia, etc., etc. Then reservoir, the source of an infectious agent. That is, reservoirs are places where the organism resides and thrives and reproduces. Example, soil, water, air, animals, elevator bottoms in people, contaminated foods, etc., etc. Humans are important reservoirs for pathogens. Carriers are infected individuals who spread the disease but do not develop symptoms. So there are some individuals that we call carriers. The person will be harboring the organism, can be transmitted to other people, but he or she himself will not develop signs and symptoms of the disease. Then portal of exit. That is the route by which microorganisms leave the reservoir. Pathogens exit their reservoirs through excretions and secretions, non-intact skins like draining wounds, respiratory tract like sneezing, coughing, and then talking, and then GIT by vomitors and then stool. Then we have mode of transmission. That is the means by which the pathogen is transferred from the reservoir, that is the carrier, to the host. These include direct contact with skin or mucous membrane, example, shaking of hands, kissing, contact with wounds, sexual contact, poor hygienic habitats, and then eating of contaminated foods, drinking of contaminated water, etc., etc. Then we have indirect contact, which also occur through fomites. Like fomites are inanimate or non-living objects that carry the agent to the host. Example, stethoscope, thermometer, bandage, scissors, clothing, linen, eating and cooking utensils, toys, money, etc., etc. So these are indirect mode of contact. We also have airborne or droplets, which are also a mode of transmission, and these include aerosols from coughing or sneezing. Then we also have vectors that can also serve as mode of transmission. These are living carriers or organisms that carry the disease agent to the host. Example, insects and arthropods. So we have a vector like mosquito that can also carry, spores or sporozoids or gametes, that is the sexual gametes of the mosquito from a person, and then you will breed it and then transmit it to another person. So mosquito is a form of a vector. Housefly is a form of a vector. Okay. Now, portal of entry. The portal of entry is the route by which pathogens enter a new host. These include the respiratory tract, the GIT, the skin, the genitourinary tract, and wounds, gastrointestinal or fecal-oral routes. Microorganisms enter the body through the same route they use to leave the body. So you might have gotten the organism through the respiratory tract. You may also give it to other people through respiratory tract, like by coughing or sneezing. And then, if it is the fecal-oral route, like you got it through the mouth, that's a fecal-oral route. It came from somebody's fetus, and then I'm talking about feces, and then you ate it. So you also pass it through the feces. So that's what we are talking about. Susceptible host. Who is a susceptible host? A host is an organism that acts as a habitat for the growth of the pathogenic organisms. Human beings who contract the pathogens are the host. Susceptible hosts are the people who are at risk of the infection. So, with the current pandemic, that is the coronavirus, we mostly say it is the elderly and those who have other underlying disease conditions or comorbidities are much more susceptible compared to other healthy individuals. Susceptible individuals include children, aged, patients with chronic diseases, immunocompromised patients, malnourished, patients with burns and on cytotoxic drugs, and also pregnant women. I didn't add that. So immunocompromised means immunosuppressed individuals, those who have their immunity suppressed, like say HIV patients and tuberculosis patients. Now, breaking the chain of infection. If you want to break the chain of infection so that you stop the infection from happening, what can we do? Let's start from the agent. Correctly cleaning, disinfecting, or sterilizing items or articles before use can actually break the chain through an agent. Performing hand hygiene, antimicrobial therapy, etc., etc. So these factors can help you to get rid of the agent. So what we're saying is, if you want to break the chain of infection, these are some of the things you can do. You clean, for instance, you are going to use a stethoscope. The organism is on that stethoscope, waiting for you to use it so that you will get it. But if you take your stethoscope, you get a gauze or a swab with spirit and you wipe it, you clean it well, you use the stethoscope, you get rid of the agent. Okay? And then antimicrobial therapy, like say you're going to use, say, some food or something, and you believe that the food may be contaminated to some extent, and then you use antimicrobial agent to wash it off. Say vegetables, wash it before you take the vegetable. You may end up getting rid of the organism, so you can break the chain through the agent. Then reservoir, breaking the chain at the point of reservoir. What do we do? Appropriate skin and oral hygiene can help take off organisms from the reservoir. Proper food storage, water treatment. So water may be contaminated, but if you treat water and the water becomes wholesome and potable, it will get rid of the agents at the reservoir level. Disposing of feces and urine in appropriate receptacles. Disposing of damp or soil linings appropriately. Changing dressings and bandages when soiled or wet. Ensuring that all fluid containers are covered or capped. Emptying suction and drainage bottles at the end of each shift or before full or according to agency policy. What about the portal of exit? Hand hygiene, disposal of wastes and contaminated linen, covering the mouth and nose when coughing or sneezing. All these factors can also terminate the transmission of the infection at the portal of exit. Now let's look at mode of transmission. Proper hand hygiene, environmental sanitation, disinfection, that is sterilization of equipment, wearing gloves when handling secretions and excretions, wearing gowns if there is danger of soiling clothing with body substances, placing discarded soil materials in moisture-proof refuse bags. That is, you want to place a discarded soil. For instance, you've changed some dressing and you want to discard it. This dressing may be moist, may be having fluid, pus and other things. You don't put it in a container that can drain and wet the ground or the floor. Others include placing discarded soil materials in moisture-proof refuse bags. So when you remove moist dressing, you don't put it in a bag that will allow drainage out on the floor. So you put it on a moisture-proof refuse bags. Then holding used bedpans steadily to prevent spillage of the content on the floor. Disposing of urine and feces in appropriate receptacles. Initiating and implementing aseptic precautions for all clients. Wearing masks and eye protection when in close contact with clients who have infections transmitted by droplets from the respiratory tract, like what is currently happening in this pandemonium. Wearing masks and eye protection when sprays of body fluid are possible. Portal of entry. Portal of entry. So we are talking about how to truncate or prevent infection or cut the chain of transmission at the portal of entry. Hand hygiene. Using sterile techniques for invasive procedures when exposing open wounds or handling dressings. Placing used disposable needles and syringes in puncture-resistant containers for disposal. Providing all clients with own personal care items, and then provision, that's what we call the PPEs. So you give people PPEs so that they can protect themselves from infections. Susceptible host. How do we help the susceptible host, or how do we terminate infection at the point of the susceptible host? Maintaining the integrity of the client's skin and mucous membrane. If the client's skin is intact, the mucous membrane is intact, the organism may fall on the skin but it will not be able to enter. Then ensuring that the client receives a balanced diet. If the client's nutritional status is strong, is healthy, then the client's immune system will always be strong, and the infectious agents may even enter but will not be able to cause infection or to cause the disease. Educating the public about importance of immunizations. So if you educate them and almost all of them or everybody is immunized, then we are not going to get people falling sick of the disease that we are immunized against. Then recognition of high-risk individuals. You recognize them and you isolate them, or you start to work with them. Then treatment. Those who are sick should be treated promptly. These will also go a long way to cut other people from getting the infections. Host defenses. Let's look at host defenses. The body's resistance to infection is determined by the state of its defensive and protective systems, which is true. Your ability to resist infections depends on the strength of your host defenses. So the defense system consists of innate immune system and acquired adaptive immune system. So we'll be taking them one by one and see what exactly they are. So let's look at the non-specific host defenses. We have physical barriers like intact skin and mucous membrane and cilia. So these are the non-specific host defenses. What do they do? If you take the intact skin, if your skin is intact, it will prevent infectious agents from entering into the system, and that is going to prevent you from getting the disease. If your mucous membrane is intact, the same thing. If your cilia is intact, we have these cilia, if you take the nostrils, we have some hair in there that we call cilia, which helps to beat out organisms that were otherwise trying to enter into the respiratory tract. If your cilia is up and working well, it will always try to beat out all infectious agents. But if your cilia is not working, then the organisms will easily enter into the respiratory tract. Then we have microbial barriers. That is normal flora. Now, normal flora, I have mentioned a few of them. I say if you take Staphylococcus aureus, it is a bacteria that can be pathogenic, but it is normal flora on the skin. It protects the skin, preventing other organisms from invading the skin. So this Staph aureus is actually a normal flora on the skin. It will make sure that it competes for space and space against other pathogenic organisms that otherwise want to come and colonize the skin. Then we have others like Escherichia coli, which are also normal flora within the intestines. or the GIT. They also colonize there, preventing other pathogenic organisms from colonizing. So they actually are very helpful in their respective areas of colonization, unless of course they are displaced or the condition changes and it's no longer favorable for them. That is when they start to cause problems or infections. Then we have chemical defenses: pH, lysosomes in tears and saliva. Now, the pH, we're talking about either acidity or alkalinity. For instance, if you take the stomach, the pH level in the stomach is very low, so it is highly acidic. As a result of these, organisms that may find themselves in food with the hope that they will get in there and start to cause problems, all of them will get killed when they get into the stomach by the high acidic medium. So that is a defensive means or a defensive method. Then lysosomes in tears and saliva are also means by which the body is able to defend itself from microorganisms. Then we have phagocytosis. These are neutrophils and macrophages. They phagocytize, that is, they engulf microorganisms that might have gotten into the body, and then they are trying to cause any infection or something. They initiate any infection. The body will send macrophages and neutrophils to them, and they will go and phagocytize these organisms, engulf them, and then destroy them or digest them, kill them. Then fever. Fever is another non-specific form of defense. This eliminates some pathogens. It eliminates some pathogens. So when your body becomes feverish, it is a form of trying to take out certain microorganisms. Then inflammatory response. Inflammatory response. That is when the invasive organisms or pathogenic organisms enters into any part of the body, and they try to start an infectious process. The body responds by what we call inflammatory process, where it sends a lot of blood to the site, sending white blood cells that will go and fight against these infectious agents. That is what we call inflammatory process. So we have looked at the non-specific host defenses. Let's look at specific host defenses. Specific host defenses involve the immune system, which responds to antigens. The specific immune response is characterized by specificity, memory, and acquired ability to detect and eliminate foreign substances. The immune response detects substances that are foreign to the body. So basically put, what I'm trying to say here about the specific host defenses is that anytime an organism, pathogenic organism, enters into the body initiates a responsive mode where it sends antibodies that actually are specific, or they target specifically the organism that has been sent into the body. So these antibodies will go and tackle and fight these antigens. But the antibodies are only specifically made for these antigens that came today. Should there be another antigen tomorrow coming in, which is different from the antigen that came, the bacteria that came in today, then the antibodies is toothless because they were not made for these new antigens. So antibodies are specifically made for specific antigens. That's what we call specific host defenses. The immune response comprises antibody-mediated and cell-mediated immunity. Antigens. What are antigens at all? I kept mentioning antigens. Antigens are foreign proteins which activate the immune response and react with the cells and chemicals to produce antibodies. So antigens could be like bacteria that has entered into the body. So they are foreign proteins, and then they will go and activate your immunity, and the immunity will now produce specific antibodies against these foreign proteins. Antibodies, they are glycoproteins that are made in response to specific antigens and react with these antigens. So antibodies, Literally, one will say antigens are enemy soldiers and antibodies are the body soldiers. So when you have, say, soldiers from a country like, say, Togo, coming to Ghana, into Ghana, wanting to kill Ghanaians, Ghana will also designate a group of soldiers to go and fight these Togolese soldiers. Then they go and fight and they overcome them, and they are stationed at the borders of Togo. So their main target is only Togo. Another time, when Ivorian soldiers are coming in, they cannot go and take those who went to fight against Togolese and they are stationed at the Togolese borders to go and fight the Ivorians. No, they will have to designate a new group of soldiers again to go and fight. So what we mean is these foreign soldiers are what we refer to. That is the Togolese soldiers, the Ivorian soldiers. They are the antigens. Whereas the soldiers that our government designated to go and fight against these foreign soldiers are what we call antibodies. So if you take it this way, it should make you to understand it easily. Now, antibodies are immunoglobulins. We have different types of immunoglobulins, and I'm going to take them and then we'll see what they are. We have immunoglobulin G. Whenever you see capital I, small g, and then another letter comes, we are talking about immunoglobulin, that thing. So immunoglobulin G, who are they? They are the smallest but the most common antibody that is found in all body fluids and is important in fighting bacteria and viral infections, and they can cross the placenta even in pregnancy to protect the fetus. So even in pregnancy, immunoglobulin G is very important, as they can cross the placenta barrier to go and protect the fetus and fight against infectious agents. So they are very, very good in fighting against bacteria and viruses, and they are the commonest in the body. Then we have immunoglobulin E. They are found in the lungs, the skin, and the mucous membranes, and they are involved in allergic reactions. So whenever your body encounters a form of allergy, immunoglobulin E is activated. Then we have immunoglobulin M. They are found in blood and lymph fluid and are the first type of antibody made in response to an infection and help in phagocytosis. So immunoglobulin E are the first type of antibodies that will be activated when any infectious agents enter into the body. And they mostly fight against these organisms by phagocytosis. Then we have immunoglobulin A. These are antibodies that are found in saliva, in tears, and blood, and protect body surfaces that are exposed to outside foreign substances. Then we have immunoglobulin D. They are also found in small amounts in the tissues that line the abdomen or the chest. How they work, as I speak to you right now, is still not very clear to scientists. So we have the cell-mediated immunity, which depends on the natural killer cells and the T lymphocytes. Immunity here can be passive or active. It can be natural or artificial, which we have already defined. So you have to go back there and see the differences between passive immunity, active immunity, natural immunity, and artificial immunity. Thank you very much for your attention.